In the last video, we talked about the different types of muscle tissue. From now on, we're going to focus on our skeletal muscle. Our objectives are to understand and describe the microscopic structure of skeletal muscle and explain the different cytoskeletal elements inside of it, actin and myosin. So a skeletal muscle fiber, the fiber is otherwise known as a cell, contains cytoskeletal elements called myofibrils. And we give special names to all the different parts of a muscle cell. So we call the plasma membrane the sarcolemma, again, sarco meaning muscle. And then just below the surface, you're going to see tons of nuclei. But the majority of the volume of the cell is taken up by myofibrils, which are bundles of cytoskeleton, each called myofilaments. And when you look at it, when scientists looked at it first under a very low power microscope, they saw that it was striped, that there were dark bands that they called A, again, the A is in dark, and light bands that they called I, the I is in light. Now, let's zoom in on that. So the myofibrils in our muscle fiber are made of contractile units called sarcomeres. They're repeating sarcomeres that are going down the length of the muscle fiber. In the middle of that I band is a dark area we call a Z disc. And the sarcomere goes from Z disc to Z disc. So when you draw it out, you want to draw Z disc to Z disc. In the middle of the A band is a lighter area called the H zone. And in the middle of the H zone is an M line. So pause this video and draw yourself a sarcomere. Z disc to Z disc. Draw the I bands and the A bands. In the middle of that A band, put an H zone. And in the middle of the H zone, put an M line. Pause this video. Do it. Draw. Okay, welcome back. Hope you had a good time drawing. Let's now zoom in again. So what are these filaments made of? The sarcomere is made of thin and thick filaments. The thick filaments are myosin, and these have head groups, and this is where we're going to use the ATP. So our muscles use a ton of ATP, and a lot of that is in the myosin head groups. They're going to split ATP to generate the power needed to contract. So the ends are dotted with these myosin heads. The thin filaments are anchored to the Z discs, and these are actin. Now, where the bands get their name, the I band, this is only has thin filaments. There are no thick filaments there. That's what makes it light. And the A band runs the length of our thick filaments. The thick one makes it dark. In the middle, when the muscle is relaxed, there are no thin filaments. So there's a little bit of open space. And what's going to happen when muscles contract, and we'll talk about this, is that the filaments are going to slide together, and that H zone is going to disappear. So we have space at rest in between our thin filaments, and when the muscle contracts, they're going to come together, bringing Z disc into Z disc. So again, add this to your drawing. Add the thick filaments and the thin filaments. Make sure you label them myosin and actin. Pause this video. Do it. Draw. Okay, welcome back. Let's now talk about how that muscle contracts now that we have our sarcomere pictures in front of us. Our learning objectives are to describe how an action potential is initiated in a muscle cell and to describe the steps of muscle cell contraction. And then we'll talk about some vocab that's important. So muscle cells are excitable, contractible, extensible, and elastic. And these words have special definitions. So excitable means the ability to receive and respond to stimuli. Contractible means that the cells can shorten when they are stimulated. Muscle cells can be stretched, which means extensible, and then they can recoil, elastic. So stretch out, extensible, recoil, elastic. For this to happen, they need to be stimulated by a motor neuron. Now, we call the motor unit the one motor neuron and all of the muscle cells it stimulates. Most of the time, a motor neuron is going to stimulate multiple muscle cells. So that forms our motor unit. And they meet at a space called the neuromuscular junction, the junction between the neuron and the muscle cell. And they're going to release little vesicles, little blebs of membrane that contain a chemical called a neurotransmitter into the gap between the neuron and the muscle cell. So they get really close to each other, but they don't touch. And we call that gap the synaptic cleft. The chemical that they release, the neurotransmitter that a motor neuron is going to release, is called acetylcholine. Okay, so take a minute, make sure you understand these vocab words that we just went over. Can you define a motor unit? Can you define a neuromuscular junction, a neurotransmitter, a synaptic cleft, and acetylcholine? So that's 5 keywords that we just covered. Write them down. Make sure you write down their definitions. Pause this video. Okay, we're going to keep going. So this is showing you again that our motor neurons come from our spinal cord. And we'll talk about that when we talk about our nervous system. and they're going to form these motor units. They're going to meet at a neuromuscular junction. The gap is the synaptic cleft, and they're going to release acetylcholine, which is a chemical, a neurotransmitter, onto the muscle cell. They come again from our spinal cord. So that motor neuron, what's the chemical they release? Acetylcholine. You'll see it abbreviated very frequently as ACH. So they're going to release ACH into the synaptic cleft. And why do they release ACH? Well, let's back up a step. So why does our motor neuron release acetylcholine? They release acetylcholine because of calcium. Remember I told you when we talked about bones that we have to keep our calcium levels really consistent, really at that homeostatic set point? That's because, in part, they're involved here at the motor neuron. So the nerve impulse, a nerve impulse is going to come from your brain. You're going to think, contract your bicep. You're going to send that impulse down your spinal cord all the way up to your arm. And that nerve impulse down the motor neuron is going to cause calcium channels to open. These are voltage-gated calcium channels. They open because of an electrical impulse, a voltage change. And calcium is going to flood into the axon terminal, into the bottom end of this motor neuron. What the calcium does is it causes the vesicles full of neurotransmitter, full of acetylcholine, to empty and release into the synaptic cleft. And then the acetylcholine is going to bind to the muscle cell plasma membrane. What happens at the muscle? So the first step again, nerve impulse comes down our motor neuron, causes calcium to come into our axon terminal. Vesicles of acetylcholine are released. Acetylcholine crosses the synaptic cleft, binds to a receptor. What does it do? It opens up these cation channels. Remember, cation means positive. And they're permissible to several ions, but the main one we want to focus on here is sodium. So sodium is going to rush into our muscle cell. And this causes a change in the electrical signal on either side of the membrane. So at rest, the muscle cell has a negative charge in it, and we're going to flood all of this positive sodium into our muscle cell. We call that a depolarization. And so the depolarization will then cause an electrical current to flow down our muscle cell. Now, how does this turn off? Well, there's an enzyme. Remember, enzymes are proteins called acetylcholinesterase in the synaptic cleft that breaks up the extra acetylcholine. This way, the single nerve will make one contraction of our muscle cell. It won't just keep going and going and going. Now, what happens when the muscle depolarizes? Well, it's the same way you can think about when you light a twig on fire. And I'm not telling you to light fires, but if you did ever light a fire, you would notice that the fire is going to move rapidly down the twig. The same thing happens to our muscle cell. That sodium depolarization is going to move rapidly down our muscle fiber. And we call that movement of charge an action potential. It's another word to add to your vocab list, action potential. Action potential, a movement of charge. That electrical charge, that action potential is going to move down our muscle fiber. So take a minute, see if you can verbalize this. How do motor neurons signal to muscle cells? Hopefully you've paused the video and come back. Now let's talk about what that depolarization does. So we know at the end, we're going to get this contraction, right? We call that sliding filament. We say muscle cells contract via the sliding filament model. So the myosin heads are going to reach up and they're going to grab onto the actin and they're going to pull them to the middle. And they're going to do this several times of attach, detach, pull, attach, detach, several times, pulling the actin into the middle, like pulling a rope. This pulling requires calcium too. Calcium is so important, isn't it? And that calcium is stored in the muscle cell in an area called the sarcoplasmic reticulum. So at rest, the myosin heads can't grab the actin because the actin has blocks. So the calcium is going to come in and it's going to release the block on actin so the myosin can grab along. And ATP is going to provide the energy to let go. So we're going to use calcium. We're going to use ATP. Calcium is going to release the block on our actin, and the ATP is going to provide the energy for the myosin heads to eventually let go of the actin so they can grab it again and pull it a little closer. Okay, let's summarize what's happening. So I want you to pause this video and see if you can explain what happens starting with the motor neuron. Motor neuron is going to get an impulse, opening voltage-gated calcium channels. Calcium is going to come into our motor neuron. That's going to cause the acetylcholine to be released into the synaptic cleft. Acetylcholine is going to bind to acetylcholine receptors, and that is going to cause a depolarization in our muscle fiber, which is going to spread down our muscle fiber. That action potential, that movement of charge, is going to open up the sarcoplasmic reticulum and release calcium into the muscle fiber. The calcium is going to open the block on action. All of a sudden, the myosin can grab and pull to the center. It will use ATP to let go and grab again. And it will repeat this process until the acetylcholinesterase breaks up the acetylcholine and we turn the signal off. Pause the video. See if you can explain those steps. See if you can draw those steps. See if you can write those steps. Then come back. Let's talk about varying a muscle's response. So we can use our muscles to do very delicate things or very strong things. And we're using the same muscle. How? Well, that's because we can contract different numbers of muscle cells. Now, a muscle cell itself either contracts or doesn't contract. We call it an all or none response. However, we can have a graded response. We can change the frequency of our stimulation. So if we just have one motor neuron firing on a muscle cell, it's going to give us a twitch. Everyone had those twitches before, where your eyes just twitching, right? That's terrible. Or your fiber just twitches a little bit. That's one motor neuron signaling onto one motor cell. However, if the frequency of contractions comes close enough, so if the acetylcholine is released quickly, those will start to sum up. And when they do, we get a sustained contraction, which we call a complete tetanus. Now, if we want to make a more forceful contraction, we're going to do that over multiple muscle cells. So we're going to recruit more and more muscle cells to contract, and that's how we get strength. So an increase in the frequency of stimulation is going to lead to a nice, smooth muscle contraction, whereas increasing the number of muscle cells involved in the contraction will increase the force. Let's talk a little bit about ATP, right? Muscles need ATP to contract. They allow that myosin to let go so we can grab again. However, we only store enough ATP in our muscles for a few seconds, and so we have to regenerate that ATP. We can do this in three ways. The first way your body does, is it will regenerate it directly. It'll take the phosphate off of creatine phosphate, which is a molecule found only in your muscles, and it will add it to ADP and make new ATP. However, you only store enough for about 15 seconds worth of contraction. So you're going to go through that pretty quickly. The other way you can do it is, if you're not taking in enough oxygen, you can do lactic acid fermentation. So you can take glucose, send it through glycolysis. This should be a review from Bio 1000. You send it through glycolysis, right? You end up with 2 ATP. And if you don't have enough oxygen, you can then convert the pyruvic acid into lactic acid and release that into your blood. However, lactic acid will promote muscle soreness, and you're going to use a lot of sugar to only get 2 ATP. You'll burn through this in about 40 seconds. So now we've taken you to almost a minute. What do we do if you want to exercise or contract that muscle for more than a minute? We gotta use oxygen. So we gotta take deep breaths. And this is the aerobic pathway. This is where 95% of your ATP comes from. Your mitochondria are going to get involved, and they're going to take that glucose, or maybe some fatty acids or some amino acids, and they're going to break it down, send it through aerobic respiration, and give us 32 ATP for every glucose molecule. And this can go hours and hours if you have enough aerobic capacity. If you're breathing deeply enough and you're getting enough oxygen into your muscles, you can exercise and use that muscle for a long time. Okay, pause this video and see if you can explain the 3 steps for ATP regeneration. In the next video, we'll quickly cover some more vocab, and then we'll talk about the gross anatomy of our muscles.